In this video, we're going to start off by discussing the various sources of energy utilized for muscle contraction, the amount of source that's available to the cell, the advantages and disadvantages of each type, and the conditions in which they might be used. And then we'll continue to talk about the two types of smooth muscle. So starting off, we have endoplasmic stores. So that is the highlight it right here. That is the ATP inside the cytoplasm. And that gets to be used really quickly in 1 to 5 seconds. It's really fast, very available, but it gets depleted quick. We'll move on to creatine phosphate. Creatine phosphate is what allows ADP to be phosphorylated with a phosphate. Okay, this just allows ATP to be more, allows it to be replenished more. And what's interesting enough too is that this creatine phosphate is found right inside the sarcomere along the medial plate, the M line, and that only lasts for 10 seconds. So, I mean, once we use up the endoplasmic storage, we move on to creatine phosphate, and we have that's 15 seconds, not very long time. Now, we'll move on to glycolysis, so that is just the breakdown of sugars, right? And again, that it's definitely a lot more. You definitely get a lot more because then we can utilize certain pumps and gradients, and we'll get, at the end of the day, we'll get 60 seconds worth of ATP or energy. Now, the best one is aerobic respiration, which uses O2, and this can last for hours. And so that's just our breathing, and we use that oxygen then to break down sugars, which then allow us to create ATP, or I guess pump hydrogen across the electron transport chain, right? And then we have ATP synthase, which allows us to create ATP. I want to start off with, before we dive into the differences between the two main types of smooth muscle, I would like to look at just the differences between skeletal and smooth. So we remember in the recent, video, that T-tubules are present only in skeletal. There's one type, and 100% of the calcium comes from the ICF in the sacroplasmic reticulum. And we also know that on the actin, or surrounding the tropomyosin, is troponin. These are also striated. Now, the difference here now is that we'll look at smooth has no T-tubules. And because it has no T-tubules, not 100% will come from the ICF. Only 50% will come from the ICF, and 50% will come from the ECF, the calcium. Now, there are two types of smooth muscle. There's a phasic contraction, there's tonic contraction. Phasic is short periods of calcium, and tonic is long periods of calcium. And we'll discuss those later just below this portion. Now, it's important to realize that smooth has no troponin and is non-striated. The no troponin is really important. That is a very key factor that, I don't know how to start this. It's very key, that has no troponin. So now let's take a look here about how we're able to make, I'll highlight the two biggest ones, how we make MLCK active and how we make MLCP active as well. So we'll first start off with this G protein. This G-coupled receptor, a ligand of some sort, like acetylcholine, a hormone, epinephrine, norepinephrine. It'll come in, it'll bind, we'll have a cascade. The alpha protein will be activated. It'll activate PLC, which then will activate IP3. Once IP3 is activated, it will open up the ranitidine channel. Once the ranitidine channel opens, then we have calcium, which will enter out, into the cytoplasm. But what's interesting here is that calcium stays close to calsequestrin. Calsequestrin is what allows the calcium not to travel very far. It keeps the calcium all relatively close. So once calcium exits the cell, it'll do two processes. It'll go this way, or it'll go this way. And I'll just focus on this one first. So it's going to exit the cell right here. It's going to come in contact with calmodulin. Calmodulin will become active. Calmodulin then will bind to MLCK inactive. Here, this is calmodulin. All the calcium's bound to it. Sorry, I need to stop this video really fast. Okay, we're good. Okay, once MLCK is active, then it can carry out the phasic or tonic phases of smooth muscle contraction. But we'll just stop there. MLCK is active. We know that process. And then we'll look over here at MLCP. Calcium, for MLCP, really, it's a little different because calcium, again, like we said, can come from the outside or the inside of the cell. We just saw it come from the inside of the cell, but we need to look at, there's also ligand-gated, mechanically gated, and DHPL type channels. Mechanically gated, as we know, is smooth muscle respond to potentially like the stretch of capillaries. Ligand, again, a hormone could come in and bind and open that calcium channel, or DHPL type from a depolarization. So if calcium comes in from the extracellular space, it'll come in right here, as we see here, calcium will actually inactivates rhinokinase. Rhinokinase is what deactivates MLCP. So if calcium deactivates rhinokinase, then we're able to make more MLCP, which essentially rips off phosphates, and MLCK adds on phosphates. So we'll look at here. So MLCK phosphorylates the light chain of myosin, so it adds on this phosphate to the light chain. Okay, now I'd like to take a look at phasic contractions. Here we have our cross-bridge cycle. We'll start off with ATP being hydrolyzed and forming ADP and phosphate. MLCK will come in and it phosphorylates, so it's going to add that blue, I'll just highlight it, that blue phosphate. Once that adds on to that light chain, as we shown here, this phosphate that was originally correlated with the ADP will pull off by that arrow right there, and it will come off. We'll perform the power stroke, but we'll still have the phosphate from MLCK on there. And then ATP will come in, it'll bind, and it'll be broken down into its counterparts, which then allows us to repeat the cycle, reset the myosin head, and allow us to contract and perform the power stroke. Now the important thing is this blue phosphate allows us to attach to actin. Because we don't have troponin in that whole process with skeletal muscle, we need this phosphate in order for us to bind to the actin and G-actin active sites. Now, the importance of MLCP is that MLCP will come in and it will actually rip off that phosphate. And that actually resets the whole process and allows us to just have contractions. So MLCK comes in, adds on a phosphate, boom. We get into the power stroke, it shifts. ATP binds. It's able to reset. Then MLCP comes in, rips it off. Once it rips it off, MLCK comes in, adds in the phosphate, and we have a contraction for smooth muscle. Now we're going to take a look at tonic. Tonic is a little bit different. Tonic contractions are, I guess, contracted for longer periods of time. I guess examples of tonic contractions would be like your sphincters, like your external anal sphincter. That is usually most of the time always closed or contracted. And I'll explain that here. So just as before, we have phosphate and ADP. ADP right there. And then, in order for us to bind to actin, MLCK comes in, adds in that phosphate there. We're able to bind to the actin and G-actin active sites. And once we're bound, right, we're bound, MLCP and ADP and phosphate all come off. So this blue one comes off, and this red one comes off, and ADP comes off. And that we just had this power stroke, but now we can't go back because we don't have a phosphate here. There's no phosphate here. Okay, and we don't have ADP or phosphate from the ADP attached. So now we're just contracted. We're just contracted. And the interesting part about this is what allows us to keep our sphincters contracted. ATP, however, will slowly bind because it has a very low affinity. So it will slowly bind, and once it'll be bouncing. It'll like add, come off, add, come off, add, come off. And once it fully binds to it, then the myosin head can hydrolyze it and break it down into ADP and phosphate. Once it's able to do this, we're able to exit what's called the latch state. The latch state is when it's in that contracted state and no phosphates is added. So once we're able to get out of the latch state, ADP will bind, resetting it, and then eventually MLCK will come in, add a phosphate, and then we'll just start that whole process all over again. Now we're going to take a look at I'm going to take a look at muscle sensory receptors, muscle spindles, and Golgi tendon organs. Okay. So basically in our muscle, we have these things called muscle spindle fibers. So here's a muscle. Oh my gosh. And inside, so you have, all this, it's very striated and stuff. Inside our muscles, we have what's called muscle spindle fibers. And muscle spindle fibers, these guys respond to stretch through the type 2A sensory fibers. Now, if a muscle stretches, it'll send a signal through the type 2A sensory motor neuron, which will then go to the spinal cord, and we'll have a contraction of sorts. Let's take a look here. I want to focus on the stretch reflex. Like I said, muscle spindle fibers, these guys here, those guys respond to stretch. Muscles, when we go to the doctor and they do the patella knee reflex, right? They'll take a hammer here and they'll smack it against the patella. Right on patella is the quadricep tendon, okay, which is where most of the muscle spindle fibers are located. And once that's hit, those muscle spindle fibers will then say, oh, we're stretching. So it's going to send through the sensory neuron. It's going to send a signal, or it's going to conduct an action potential. Once it gets down there, the sensory neuron, or this type 2 sensory fiber, will synapse with the alpha motor neuron and interneuron. So this red is the interneuron, and the alpha motor neuron is this green. Okay, so now that synapses with those two, we're going to have two things that happen. In order for our leg to kick up, okay, our quadriceps need to contract, and our hamstrings need to relax. So the alpha motor neuron to the quadriceps is stimulated, while the alpha motor neuron to the hamstring is inhibited. And so we'll have the net movement of a extension. Okay, now we'll take a look here at the golgi tendon organ, which is basically a safety mechanism that does the opposite of the stretch reflex. Okay, so if it does the opposite of the stretch reflex, that means we're going to do flexion. So that means we'll go this direction. However, it's really interesting. Golgi tendon, so right here on the tendon, the golgi tendon senses excessive tension. So usually the golgi tendon reflex allows us to protect our bones from being ripped apart by our muscles. Essentially it. So if there's excessive tension, if we're extending really hard, the golgi tendon organ will actually combat that response and force the leg to go into flexion. So take a look. So here we go. Right here, it senses excessive tension. So we're going to go send it through the sensory neuron. Again, it's going to connect an action potential. Once it reaches the spinal cord, the sensory neuron is going to synapse with this interneuron, and as well as the alpha motor neurons. However, the alpha motor neuron to the quadriceps is inhibited, while the alpha neuron to the hamstring is stimulated. So it allows us to go into the flexion state. And this is not what we're supposed to be talking about, but I really want to add it in because I think it's really interesting. So if we step on attack, right, our initial response is going to be to lift our leg, right? But that means we need to stabilize our other leg. So that's called the cross extensor reflex. It's stimulated after the withdrawal reflex and stimulates the extensor muscles on the opposite side. Okay, so the sensory motor neuron goes up, synapses, right, in the spinal cord, synapses there. Okay, with the withdrawal reflex right here, this is our withdrawal reflex. Okay, it's going to synapse with the interneurons and the alpha motor neuron. It's going to send a signal to the extensor to relax and the flexor to contract. This motion would allow us to lift our leg, but because we lift our leg, we need to be supported by this other leg. And so what's going to happen is the same signal from the sensory motor neuron, it's going to tell the extensors on the opposite leg to contract, right? So your quadriceps to contract, and then your hamstrings to relax so that you can stabilize yourself. So basically, that's all we have for today in this video. Hopefully you found that interesting and you were able to learn a lot. Thank you for joining me.